Hey, my name is Julian Ginori, and I'm a PhD student in the Environmental Horticulture Department at the University of Florida. I'm here to talk to you today about tissue culture and micropropagation, so you can decide if it's something worth pursuing in your research or in your business.
The first concept I wanted to cover are the two terms micropropagation and tissue culture. These two terms are often used interchangeably because from a superficial point of view, they both follow the same stages. Where these two terms differ is perhaps in their purpose or use. Micropropagation places a large emphasis on the ability to produce hundreds to thousands of clonal plants for, most often than not, eventual sale. Tissue culture, on the other hand, aims to keep plantlets on media for eventual transgenic studies via transformation through agrobacterium or other methods. Tissue culture is sometimes referred to as the beginning stages of micropropagation. Speaking of stages, this video is going to cover all the stages of tissue culture and micropropagation. If we were to visualize the stages, tissue culture consists of initiation, multiplication, and rooting. Micropropagation takes it a step further and includes acclimatization. Going back to the point I mentioned earlier, micropropagation is broken down into two stages, tissue culture and acclimatization. Prior to these stages, I like to include stage 0, which is selection and preparation. This is a step where you define what your goals are, determine what plant material and media formulas you'll use, and prepare your working area. Good planning in stage 0 leads to higher success in future stages. Now that we know all the stages, let's go into more detail of each. Stage 0, the planning stage. Prior to introducing any plant to tissue culture, a review of the literature is essential. This one step can save you weeks of work and hundreds of dollars in chemicals and other supplies.
Tissue culture is all about minor adjustments to your media formulas and plant material. So if someone's already published a peer-reviewed article on your species of interest or close relative, this could allow you to hone in your media formulas before ever even trying it yourself. The same can be said for plant material.
In its simplest form, tissue culture is about introducing a small fragment of your plant, called the explant, into an artificial medium that contains nutrients needed for growth. The different types of explants you can use include seeds, embryos, organs, and more. Most commonly used explants include leaves, shoots, and roots from which calyces are induced.
Finding out which plant material works best for your species is key to an efficient tissue culture pipeline. But that's half the battle. The other half is determining your media formulas.
Each stage of the tissue culture process will require different media formulas containing various concentrations of nutrients, hormones, and antibiotics.
If you have a game plan for what media formulas you're going to test in stage 0, the prep stage, that's going to enable you to find the optimal plant media mixture for your species much faster.
Now that you've locked down the plant material you're going to use and the media formulas you're going to test, the next important step is preparing your work environment. I cannot stress enough how important a sterile work environment is to the success of introducing plant species to tissue culture. Proper PPE is a must, and the laminar flow hood should be sterilized prior to each use, along with all the hand tools and other supplies you will be using. Okay, with all that prepped, we are now ready for stage 1, the initiation stage. Once your plant material is collected, it must be sterilized. The plant material is typically run under water to remove any loose debris, followed by a 70% ethanol wash. This is followed by a rinse with sterile autoclaved water and then a bleach wash. Depending on what type of plant material you're using and your species, the concentration of bleach varies, but a good starting point is 10% and going up from there until you find the most suitable concentration for your species. It is important to follow the bleach wash with multiple steps of sterile water rinses.
I'd like to point out that there's quite a bit of variation in the sterilization process. Some plants may require multiple ethanol or bleach washes. Others may require longer or shorter durations of these washes. It's up to you as a scientist to determine the optimal protocol for your species.
At this point, the sterilized plant material is ready to be transferred to your pre-made media. There is significant variation in what components go into your media, and this changes from species to species and for each of the different stages. Broadly speaking, these components include water, gelling agents, nutrient salts, vitamins, amino acids, carbs, and plant growth regulators. A good starting point would be a simple MS media, which stands for Morishige and Scoob. This is pre-made mixture that includes all of the macro and micronutrients that a typical plant requires for tissue culture. From here, you can add other components that I mentioned earlier, such as carbohydrates and gelling agents. Plant material prepared for tissue culture often cannot produce their own carbohydrates, which is why the addition of sugars is so important. An equally important component is your gelling agent. This is what will turn your media solution from a liquid into a semi-solid, jello-like substance, perfect for your plant material choice to sit on and eventually root in. The most common gelling agent is agar, but for situations where the yellow tint could become an issue, there are completely clear gelling agents such as phytogel that produce a colorless gel. Higher concentrations of gelling agents will result in a firm media, while the opposite is true for lower concentrations. A consistent theme that you'll hear me mention time and time again is the need to test multiple components and concentrations to find the one that works best for your species. In order to properly homogenize the media solution, it will either have to be autoclaved or microwaved and allowed to cool slightly before plating in a petri dish. Once cooled, you can carefully pour the media into the petri dishes. These come in multiple different sizes and depths to suit all of your needs. Scientists can also vary the quantity of media that is poured into each of these dishes. Typically, you have light, medium, and heavy pours, which can be used depending on the stage of the process you are on. For plant material that is just being introduced to media, a light pour is recommended, as you'll most likely be cycling out plant material as contamination occurs. A benefit of the lighter pours is less wasted material, which over time can end up saving labs hundreds or thousands of dollars in materials cost. Once the media has been made, plated in the petri dishes, and allowed to solidify, it is time to transfer your plant material to the petri dishes. Even with great care and a lengthy sterilization process, it is still possible to find contamination in your samples. The last step of stage 1 is ensuring all of your samples are sterile. This involves constantly checking up on your material for signs of contamination and subbing out the clean plant material to new petri dishes as soon as possible to avoid cross-contamination. You may have to go through multiple cycles of subbing before you are finally left with sterile plant material. Every time an explant is moved to a new petri dish, that dish should be sealed with a simple saran wrap to lock in the moisture and keep out contamination. Now that we have sterile material, from here on out termed explants, it is time for stage 2, multiplication. Compared to stage 0 and stage 1, this stage is pretty straightforward. This is a stage where we modify our media to promote shoot development. It is worth noting here that the development of roots is not important in this stage because all of the nutrients are provided for the explants in the media, and we're going to be excising the newly developed shoots to new petri dishes. As stated in the title of this stage, the name of the game is multiplication. We want to produce as many explants as needed in a short amount of time and utilizing as little space as possible. This is a true highlight of tissue culture and micropropagation, the ability to produce hundreds or thousands of clonal explants in a very small space compared to something like shoot cuttings in the greenhouse. Shoot regeneration is induced using plant growth regulators. Cytokinin is a good option to promote the growth of shoots, but once more, you need to test the best concentrations for your species. Once the explants have been multiplied, we are ready to begin stage 3, rooting. It is time to turn these explants into fully functioning plantlets with their own shoots and roots. The explants will first have to be transferred to a new media supplemented with auxins to induce rooting in the regenerated shoots. Certain species have more trouble than others in rooting, which is why multiple concentrations of varying auxins need to be tested to find the one that suits your species the best. After a few weeks, new roots will form, and it will finally be time to move on to stage 4. Stage 4 is acclimatization. I would like to point out that we are no longer in the realm of tissue culture. We have now transitioned to the final step in micropropagation.
This is the home stretch, where we slowly prepare these plantlets for life outdoors where they belong. It's important to note that up to this point, these plantlets have been growing in completely sterile conditions, with all the nutrients they need and environmental conditions met. They have had no instances of disease or pest, and thus are extremely fragile.
Ideally, you want to modify the environmental conditions one at a time. Plantlets should first be transferred to a softer soil, avoiding larger pieces of substrate like bark, as these could impact the plantlet's ability to survive. Some scientists prefer to autoclave their soil in the beginning stages to reduce the exposure of plantlets to pests or diseases too early in the process. Once in soil, the plantlets should be kept in a controlled environment room under a humidity dome and allowed to adapt to their new substrate. Humidity is key during this stage. These plantlets have been in a very high humidity environment their entire lives and thus need to slowly be weaned off of it to more natural humidity levels. This process can take 1 or 2 weeks depending on the species you are working with. Once the plantlets have grown into their soil and adapted to a lower humidity environment, they are ready to be transferred to the greenhouse for continued growth. At this point, the plantlets are almost ready for use. They should be allowed to grow into their pots and fully acclimatize prior to their final use, or you risk high mortality rates or misleading data in studies. To wrap up the entire micropropagation process, we started with a species of interest. We determined what plant material was best to use for tissue culture. We sterilized the plant material and transferred it to tissue culture media. We turned a few pieces of the plant material into hundreds or thousands of explants through multiplication of regenerated shoots. We then turned those explants into plantlets by inducing rooting, and then we finally prepared those plantlets for the real world by slowly acclimatizing them.
There are many moving parts when it comes to micropropagation, but once a good protocol is in place, it allows for the production of hundreds to thousands of clonal plants in limited space and a short amount of time. And that is the true beauty of micropropagation. Thank you for watching this video.